Hello. Good afternoon, everyone. I am Mamta Sawant from ICMR NIRRH. I welcome you all again for session 2 of the Mini-Symposium. So the first speaker for session 2 we have is Dr. Debjani Saha from Premas Life Sciences. I will begin the session by introducing her to you. Okay. So Dr. Debjani Saha is currently working as a group product manager at Premas Life Sciences Private Limited. She has done her PhD in molecular genetics from the Indian Institute of Science, where she investigated the pre-mRNA splicing mechanism with S. cerevisiae as a model organism. She has worked briefly in University of Manchester with Dr. Ray to dissect the functional utility of second step factors. She is involved in technical marketing of genomic portfolio, which includes technologies like next generation sequencing, single cell genomics, spatial transcriptomics, and many more. Dr. Debjani, I would request you to start your talk.
Thank you so much, Mamta, for the kind introduction. And for the benefit of the bandwidth, I'm not turning on my video because, networks can be fickle. But my today's presentation, is basically delving on the clinical applications of next generation sequencing and also implications on single cell genomics. So normally we would actually classify single cell genomics as a means to get to an end where you can actually answer complex questions, but they're mostly research focused. But we also have a huge number of clinical implications of single cell genomics, and we'll delve through a few examples on those. Right. Just to introduce, before I go to introduction of, Illumina, next generation sequencing, etc., I'll just introduce Premas a little bit. I'm sure my colleague Nitya would have already spoken about Premas, but just in a couple of lines. Hello?
Slides. You're not able to...
You're not able to see the slide, is it?
Yeah.
Hang on, just 1 second. I'm trying to... Oh, okay. Share screen. Can you see my slides now? Are my slides visible?
Yes, ma'am.
Yeah.
Okay, great. Thanks. So just to introduce Premas, we were incepted over 15 years back as a contract research organization, and that arm of ours is still alive and kicking. And, our firm is currently engaged in basically working on an oral vaccine for COVID. That's the latest work that they're doing. Premas Life Sciences, on the other hand, is involved in reaching technical, a lot of advanced technologies to research labs and diagnostic setups, right? And as far as the technologies we represent, among them is next generation sequencing from the Illumina tables, which basically has over 90% of the world's sequencing share and has a bunch of instruments across throughputs. So we have benchtop sequencers, we have mid-level sequencers like the NextSeq, and we have the NovaSeq, which is basically the retail scale sequencer for genomes. And basically, like, sequencing as a technology spans a variety of applications, and these applications are very familiar to all the researchers who are probably in this meeting and beyond, because it's basically helping addressing a lot of important questions. The first application that I would like to focus on in terms of clinical implications is oncology. And we all know that, our genome is dynamic, it's not static. And when you're actually trying to study cancer, one snapshot is not enough. because we have different kinds of mutations which can actually lead to cancers. Among them are those which you're born with, which are germline, and then you have somatic, which are mutations you accumulate along your lifetime, and those basically lead, may lead to cancers. And also, to focus on oncology, the world and across several countries have formed these groups and consortia where they've taken up different tissues and they've taken up different organs, and they've studied the cancers associated with those tissues. And through that information, today we're sitting on a huge reservoir of information across tissues, across various mutations, and across the various prognosis and diagnostic outcomes, right? And these basically have helped us in a variety of functions, and to stratify the risk, assess the risk for early screening and detection, for diagnosis of the cancers and characterization of the cancers, for therapeutics or theranostics, and for monitoring how well those therapeutics are working, and also to check whether or not the relapses are occurring. Uh, Typical challenges in diagnostic settings, as far as oncology is concerned, is we all know, because cancer is a disease of the genome, you essentially have to test a lot of genes, and testing today is iterative. I mean, testing in a lot of settings is iterative. Essentially, you go from one gene to another gene to another gene, and all these single gene tests end up costing a lot of time, money, and also the tissue burden that you need to do all of these is pretty high, right? Whereas when you actually move to multi-gene panels, as is enabled by next generation sequencing, and the prime reason why that is possible is because of the scale of next generation sequencing and the small time taken for addressing all of these things is very less, right? So over the past many years, when sequencing prices have dropped, that has really enabled us to bundle in a lot of genes, and basically it's basically helped us to study these amalgamated gene panels, which basically save on time, tissue, and also it's not iterative, so you're getting a lot of answers within the same panel, right? Now, as far as precision medicine, so all of this is basically, converging into a bigger branch of science, which is basically known as precision medicine, right? And precision medicine is where you're actually tailoring the treatment for individuals rather than having like a one-stop solution for everyone, right? Now, as we all know, in terms of any kind of therapy, and that is something which specifically fits with oncology, but is slowly migrating into other disciplines also, that one size does not fit all, right? And even if you take something as basic as chemotherapy or radiotherapy, there are responders, there are non-responders, and essentially you cannot uh, basically tailor a treatment which can give the same response for everyone. And the way most clinical trials are designed are, can be broadly fit into two categories. One is the basket trials and one is the umbrella trial. And what are these two, right? The umbrella trial is essentially when a single cancer type is treated with various therapeutic agents, right? So when you have, say, somebody has lung cancer as a general outcome, right? That lung cancer can be caused because of various mutations. And the molecular reasons, behind that lung cancer can be multiple, right? So here you have a single cancer type, which is being caused by multiple molecular abnormalities. And, you have a bunch of drugs which can be used for targeting all of these abnormalities. On the other hand, then you have the basket trials, wherein you have the same molecular abnormalities across the various tissues causing different kinds of cancers. and you have a single drug which can actually help target these abnormalities across the various tumor types or cancer types, right? So there's a single therapeutic agent, there's a single anomaly, but there are potentially many outcomes. And all of this, again, has been feasible because of the knowledge of the genome or because of the characterization of the genomes, right? So you have multiple therapies targeted to different individuals, and the way or the reason why we're able to do it is because of the knowledge that we have from their respective gene targets, right? Just coming to the commercial part of it, Illumina has a bunch of panels, and aside of that, because there's an ecosystem created, you have a lot of companies which have their panels which are tailored to the Illumina sequencing. Right? From oncology, if you just move to rare diseases, when you actually talk about a disease being rare, then the numbers that you're normally talking are about anywhere between 1 in 1500 to 1 in 2000, right? But assuming that there are billions of people living worldwide, and 1.3 billion people living in India itself, that number is also huge. And there are about 7,000 rare diseases that we know that are globally recognized, and so many others which are not even characterized very well, right? And as far as the burden in India is concerned, this is a chart which kind of roughly gives the Indian burden of rare diseases, right? And the fact that, not only is it physically grueling, but it also has a social and an emotional and a financial implication is something that, a family who has a rare disease, a patient is very well versed. So there's a huge diagnostic odyssey that a person goes through before the correct diagnosis is reached at, and that includes a huge amount of time, a lot of money, etc. Right? So genome sequencing, exome sequencing has really helped us address this specific challenge. And this very nice work from IGIP, I've just pulled it up, just for an example, is a beautiful illustration of how exome sequencing led, to an investigation and interrogation of this particular disease, where so it's basically lamellar ichthyosis, and the phenotype is, hyperpigmented fish-like scales all over the body, loss of eyebrows, alopecia of the scalp margins, heat. A high heat sensitivity, photosensitivity, etc. What they did was they did exome sequencing. They have this consortium called the Guardian Consortium, wherein they have basically tie-ups with a huge number of clinicians all over the country, who send them samples which are difficult to diagnose. And this was one of the cases where a set of boys, a couple of boys from the same family, had this predisposition, and they saw that there was a mutation in the transglutaminase gene, which was then resolved by exome sequencing. And identification of the mutation helped in the prenatal testing of the mother the next time that she was pregnant. And when they actually figured that the child was okay through pre-implantation genetic genetic diagnosis, and then they had a child with a normal skin, right? And also for the twins, which the boys which were affected, they gave an administration of retinoic acid at a very low dose over 2 years, and they could alleviate some of the symptoms. So it's a nice therapeutic intervention that they could plan because they knew the molecular outcome of the disease, right? And to help in all of this, you have a bunch of databases which can actually give you a lot of information about the genes that are implicated, and also the fact that we have specific cloud-based and server-based solutions which can really help in very fast analysis of even complicated data from genomes and exomes, right? Moving over to some examples of clinical implications of single cell genomics. The first example that I want to show you, and Nitya has spoken about, single cell applications, which can be interrogated using the 10x genomics technology and Illumina sequencing. So what I'm going to outline is some nice and examples of how this can be actually taken to real life situations. Like I said, most of us think of single cell genomics as a means to basically tease out complex questions and don't think of it as something which might have a direct clinical implication. But this nice study from Aviv Rajav's lab shows that no, it can actually have a potential clinical implication. So here, what they did was they studied mouse tracheal cells, and they did a single cell, single cell RNA sequencing of mouse tracheal cells. And what they saw was that there was this very small subset of cells, actually, which is present in less than 1% frequency, which was basically expressing CFTR, right? Now, in previous studies where this kind of a scale was not possible, this population would have been missed. And so most of the people who were targeting gene therapy for cystic fibrosis were targeting the ciliated cells of the lung. But here, because they could, pull out this rare cell population, which is less than 1% of the total cell population, they could figure out that this was the particular cell population which was responsible for the expression of the protein, and hence could be a good therapeutic target. And this was only possible because they were able to, they were able to analyze a lot of cells with the technology, right? Similarly, there's this another example, and this is also a very, very nice example of mechanisms of immunotherapy resistance. Okay, this is from Professor Oud Chapuis's lab, and so this is a classic case. This is a case where this woman had Merkel cell carcinoma. Right now, this is an aggressive kind of skin cancer, and she was given an infusion of T cells which could recognize the oncoprotein. Right, the oncoprotein has a jargon which is really not important. But what was really stark in observation was that this lady responded to the treatment, the immunotherapy treatment, for about a year and a half, and then she became resistant. Like, she stopped responding, right? And there was a resistance acquired, right? Now, this kind of immunotherapy resistance is not something which is unheard of. It's fairly common, right, in immunotherapy. And one of the reasons why it's, patients often become resistant to immunotherapy is because of the depletion of the T cell, CD8 plus T cell population, right, which is the prime responder here. When they actually did the single cell, analysis of the cells, what they saw was the CD8 plus cytotoxic T cells were there, right? It wasn't that this cell population had depleted. So then what was going on? And they also did a further mutation analysis, and they saw that there was no DNA mutation that can be ascribed for this resistance, right? Now, so what they saw instead was that the cells, expressing HLA-B, so HLA-B was being down-regulated in the cells, in the T cells population, right? So now the T cells were there. They were able to recognize the antigen, but because of the loss of HLA-B, and that's what they saw, they were not able to present the antigen properly, right? So because of that, the patient became immunotherapy resistant. And this was a reversible process. This could be reversed. So, and this was not basically specific to Merkel cell carcinoma. This is something which can be extrapolated to other cancers also. And because this was not previously investigated in such depths, and when you do IHC, you do a pan HLA-ABC IFC, so there's no way to figure out the downregulation of HLA-B. So because of that, maybe this mechanism was underappreciated until this thing was done. So this is a nice example, and this can potentially be extrapolated to other cancer types, so on and so forth. Another example, in a diametrically opposite disease, and that is Alzheimer's disease, is where the researchers from Broad and Harvard, they studied about 80,000 nuclei from about 48 individuals, right, along various severities of Alzheimer's disease, right. So what they did was they did single cell RNA-seq and 3' gene expression, and essentially they saw this t-SNE plots, and they could actually delineate these 6 major types of cells across which they studied the gene expression. So if you just see this heat map here, you can see that this particular column on the right basically shows the bulk gene expression, and this looks nothing like the gene expression in the 6 different kinds of cells. So the 6 cells that they took were oligodendrocytes, excitatory neurons, inhibitory neurons, astrocytes, oligodendrocytes, precursors, microglia, etc. So these 6 cellular types, the gene expression, when they teased it out, the gene expression looked nothing like the bulk gene expression that they also did in a parallel experiment, right? Which just showed that a differential gene expression happens in different kinds of cells, and the essence of it cannot be captured at all by a bulk gene expression analysis, right? And when they studied further, and that is not present in this slide, but when they studied further, they could figure out that in all of these 6 cell types, right, initially the genes which are related to Alzheimer's disease pathology had a very, very different kind of a gene expression, and that you can see here. But towards the later stages, the gene expression became, little common, right? So there wasn't so much of a stark difference between the gene expression in the later stages, because there it was basically global stress response genes that were being expressed. But in the initial stages, the 6 cell types had very, very different kind of gene expression profiles, and those were related to the Alzheimer's disease pathology, right? So nice example. Since we're talking so much about COVID, I thought it was really necessary to probably include COVID kind of work, and this was basically just to show the macrophage. So this is again from this university in China. I forget the name, but this was published in Nature Medicine, and this was basically showing the differential cell population or gene expression in healthy controls versus mild cases of COVID versus severe cases of COVID, right? So what we have to do is basically correlate the plots on the right and the violent plots on the left. So the schematic, not schematic, the plot on the right clearly shows that the cell composition between the healthy control, the mild control, and the severe category patients is very, very different, right? So essentially, this is basically the UMAP plot, and this is the violent plot. And what they basically focused on was 3 different kinds of cells, right? The FCN1 positive cells, which is the vinculin 1 positive cells, the secreted phosphoprotein 1 cells, right? And the fatty acid binding protein 4 cells, right? And these are, viral plots for all of these 3 markers. And as you can see, the, the expression of the various markers in the healthy controls versus the mild versus the severe category is very different. So the healthy control and the mild patients both express the FABP markers, right? And the severe category, in the severe category, the FABP positive cells are almost missing, right? So here, as you can see, in the severe category of patients, the FABP plus markers are completely missing, marker cells are completely missing. And the FCN1 positive and the SPP1 positive cells are, increased or are deregulated, dysregulated, right? And those are the cells which are basically involved in cytokine storm, which are typically associated with severe category patient. So it's showing a dysregulation of the highly inflammatory chemokine producing macrophage subset of cells in the severe category of patients through the interrogation of these 3 major subsets of cells, right? Also, what can be easily seen here is that the T cell population, clonal population, increases, the total T cell population and the clonal population increases in the mild category of patients, whereas in the severe category of patients, the expansion is not that much, and that's what it's also represented in the right side diagram. As and the way that you can extrapolate these results is that the adaptive immunity basically springs to the rescue in mild category of patients, and that is explained by the clonal expansion of the T cell population, and that is seen in the healthy controls and severe, healthy patients and severe in the mild patients, and not the severe category of patients where the clonal expansion of the T cell population is much lesser. Finally, this work basically is from Yutaka Suzuki's group from the University of Tokyo. And here he studied ductal carcinoma, in situ of the breast. And in the left, you have the H&E staining of the cells, wherein they've categorized these 3, clusters of cancer, and these 4 clusters of healthy cells, which basically refer to the tumor microenvironment. So these 3 clusters refer to about 400 plus spot. This is basically a Visium, this is an example of the Visium technology, wherein you were trying to do an in situ, a spatial transcriptomics of the various cells. So here, as you can see, what they're trying to study is the GATA3 mutation, right? Now, GATA3 mutation is associated with very poor prognosis of breast cancer. And what they tried to see was basically they tried to analyze these sets of genes, which could be ascribed into 3 categories. One was the epithelial mesenchymal transition. Second was angiogenesis, and third was estrogen response, right? And what they saw was the green color is the spots where the GATA3 mutation is not present. Now, this is a mutation which is basically localized in 3' end of the transcript. So, it's retained in the library prep, so it's easily studied through sequencing. So in the green regions are where the mutation is not there, and the red regions are places where the mutation is there. And what can be seen, and you can actually ascribe the color to the gene categories, that the genes associated with epithelial mesenchymal transition and angiogenesis were upregulated in the cells which were GATA3, which had a GATA3 mutation, right? And the one where you had a estrogen response, the estrogen response, linked genes were upregulated in the cells without the mutation. So where does it translate into the clinical implication? Now, the way you can translate it into a clinical implication is that many of the ductal carcinoma cells do not progress into the invasive category. So often the therapy administered is, an overkill, really. But we don't really have too many markers which can actually delineate the cells which do progress into the invasive category versus those which do not, right? With spatial transcriptomics, with technologies like single cell gene expression, we are able to get much more information about these markers and their specific localization in cells, and hence you can target therapies which are probably more conducive to the kind of tumors or to the kind of cancers that exist. Right? So, just to kind of summarize everything, single cell sequencing essentially can basically give you valuable information across the various disciplines, be it immune system, be it the digestive system, urinary system, cancers, microorganisms, host pathogen response, the nervous system, reproductive system, etc. So you can actually have a dissection of problem across various disciplines, and this can really help you lead to better therapeutic outcomes, and this will actually gain much more momentum as we gain much more knowledge, and we're able to apply it to clinical scenarios. um, that is pretty much that I wanted to say in today's presentation. Thank you so much for your attention, and I'll be happy to answer any questions that you may have. Thank you.
We'll take the question answer sessions in the discussion part.
Sure. Thanks, Mamta. Thank you, and thanks everyone for listening.
The next speaker for the day is Dr. Sunil Kumar Raghav. He is currently Scientist E and Group Leader at Institute of Life Sciences, Orissa. He has completed his PhD in Biotechnology from Institute of Genomics and Integrative Biology. His postdoctoral work was on understanding molecular and epigenetic mechanisms of human T helper cell differentiation from the Molecular Immunology Group, Finland. He has more than 50 peer-reviewed publications in international journals, including Cell, Science, and Nature. His group has recently identified interesting molecular targets in dendritic cells that could be targeted to perturb T cell function and explore its potential in therapeutics. His lab works on employing functional genomics approach to characterize the transcriptional control of immune response generation. Dr. Raghav, the floor is all yours now. Thank you.
Thank you, Mamta, for the introduction. And yeah, I can share the screen. Is it visible?
Yeah.
And I'm audible as well, huh?
Yes, sir.
Okay, thanks a lot for the nice introduction. And thanks, Deepak, for organizing this symposium and inviting me for giving a brief overview of what we are doing on single cells. And currently, anyway, we are working on COVID-19 from last 1.5 years and trying to do other things slowly because everything is like slowed down. So that's really good to discuss at least few things. Okay, just a second. Okay, so it's full screen now.
Yes.
Okay, so today I'm going to talk about one of the interesting work that we did recently. And this is basically because last year we were very much interested to understand why some of the individuals, they don't contract disease, they don't contract infection, even if they are in the same family or nearby and they were not contracting infection. But this time it's definitely changed. It appears that if somebody is infected in family, then definitely all are infected because the infectivity of the virus is drastically increased. So in this talk, I would like to tell you about this genomic approach, where we try to identify if there is any protective immune signature in these direct contacts of the infected patients. Do we see any signature? And if their cellular immune response, like in terms of B and T cells, there is some signature that is coming in PCRs or BCRs. So that was the idea to do this work using a cohort that we selected. So before going into this, I will just give you some brief introduction because I think it's important to give a brief introduction about like immune response and how it's generated in SARS-CoV-2 and why currently we see that the infection rate is so high. What is happening actually? And then I will take you or walk you through this results. So we very well know that this SARS-CoV-2 virus, when it infects through our nasal and oral cavity, and it resides in the nasopharynx area. So what happens in the nasopharynx area when it multiplies? It's encountered by our antigen presenting cells. One of those cells are dendritic cells, and my group basically works on dendritic cells in different aspects, a lot of different things. And we try to manipulate the dendritic cell basically in our lab to control these CD4 or CD8 T cells. But what happens in case of SARS-CoV-2? When the virus load is high, then these antigen presenting cells, they basically engulf those viruses, and then they present them their peptides to CD4 T cells. We call them T helper cells. So some of the CD4 T helper cells, they have already the receptors, TCRs, that can cross present with the MSC2 presenting SARS-CoV-2 antigens, antigenic peptides, and those cells undergo strong clonal expansion. Clonal expansions means they start multiplying response. And then these cells also activate B cells in our body, spleen basically, secondary lymph nodes. And there they activate B cells, and these B cells also undergo clonal expansion. But specifically those cells which can recognize those antigens. On the other side, these antigen presenting cells also cross present through their MSC1 these antigenic peptides to CD8 T cells. We call them cytotoxic T cells. And once they recognize these peptides, they also undergo clonal expansion heavily. But then in your body, you have a large number of cells that are recognizing the antigenic peptides. And there are some cells which are basically non-specifically present there, CD8 T cells and CD4 T cells. Similarly, the B cells. So you have a big number of these B and T cells inside your body. These clonally expanded CD8 T cells, they start secreting these perforin granzyme, which are very important molecules for killing the virus infected cells, and by other mechanisms. They secrete a lot of inflammatory cytokines, these CD4 T cells, as well as these CD8 T cells. And if this cytokine storm is uncontrolled, that leads further to damage of lungs and other organs. And that is happening in a lot of cases, that the inflammation is uncontrolled. But what happens when these infection is controlled in a like a controlled manner? You have good amount of B cells, CD4 T cells, and CD8 T cells. They control or contract this infection, and then the SARS-CoV-2 is cleared. Generally, it's done in 5 to 10 days. But if hyperinflammation is there, then it can create problem. So what happens? I would just explain a little bit about why in the recent scenario, we see that these antigenic peptides or immune escape variants are arising, the infectivity is high. So I will take you a little bit to the genetic part also, because that will help you to understand what are the immunogenic peptides and how the immune escape is happening. So as I told you that this is the virus. Okay, so we know that these spike protein, everybody know now, I think the spike protein. So these are the flower-like structures. And this is the structural, like a 3D structure, I would say it's more like explaining the different domains of this spike protein. So there are two domains, S1 and S2, and there is a receptor interacting domain, ACE2 receptor, which basically leads to the internalization of this virus inside the host cells. So this spike protein, as we have heard of, this is very important for generation of antibodies, because strong immunogenic peptides are present on the receptor interacting domain, as well as the S1 and S2 domain. So that's the reason that the virus is adapting so much within this spike genomic region, leading to this adaptation or immune escape variants. So here is just shown that these are the antigenic peptides, which are these train basically CD4 T cells to educate B cells, and then these B cells secrete a large amount of antibodies against these antigenic peptides and neutralization of these viruses through binding of this spike protein or coagulation of this. Why this is happening? Because this is very recent slide, because my group is also involved in this genomic surveillance of COVID virus in those 10 institutes. So what is happening here, this in the left side, I am showing you that this is the Indian variant that is recognized basically in February and on March in Maharashtra. And these are the two major mutations that were identified. This is the receptor binding domain, as I told you. So this L542R, this mutation basically leads to change in the antigenic peptide of this receptor binding domain, and that leads to immune escape variant of this virus. And with time, what happened that these regions are further mutated. So there is like a 617.1 variant, 617.2, and 617.3. These are basically accumulation of more variants along with these two variants in these viruses. And recently, if you can see here on the right side in this plot, in April end, you can see that this 617.2 is like prevalent. It's like 90% of the virus is 617.2. And this is the virus which has very strong immune escape variants, and this increases the binding affinity with the S2. So I just wanted to say that the immunogenic peptides that are cross-presented by dendritic cells or antigen-presenting cells to your CD4 and CD8 T cells are very important to generate a clonal expansion of these immune response cells to contract the infection, or basically remove these virus-infected cells or the virus bodies. So coming to the main part, like why we decided and what was the idea behind to work on this cohort or a small project. So here we identified the direct contacts ever. Somehow I'm getting this here and then I need to put it. Okay. So we identified the direct contacts of these COVID-19 infected cases. They have some specific protective immune signature. The reason was, I told you that somehow some individuals are protected, where others are susceptible to it. So what we looked here is, we looked into the antibody titers in contacts versus their infected cases, parallel. We looked into the cytokine profiles. So we basically use 48 plex cytokine profile to identify what cytokine signature is there in the contacts versus patients. So if we can see some cytokine signature which is prevalent in contacts, how is the BCR and TCR response or B cell receptor or T cell receptor profile? Do we see some signature of B cell receptor or T cell receptor that is enriched in the B cells or T cells that are clonally expanded in context versus the cases? So in the study plan, what we did, because this need to be controlled study, because here we are looking into the direct context of the infected cases. So how you define the direct context? So according to the WHO, the direct contexts were that were basically for long time uh, within 1 meter distance of the infected case, and they should be there, and then you can call them direct contacts. So in this study, what we did, we included 47 individuals, and these 47 individuals, they migrated from Gujarat to Odisha, to the Berhampur region, Ganjam region basically, and they were quarantined directly, because that time the quarantine was going on when they were arriving here. So we took these into a study, and from these, what happened, that after the mild symptoms, mild to moderate symptoms, those individuals were shifted to the hospital, whereas their direct contacts were in the quarantine center only, because they didn't have any contact, they were not PCR positive. So we selected these two groups, PCR positive contacts, PCR positive, and their direct contacts, around 23, 24 individuals in both. And then PCR positives, symptomatic and asymptomatic. This is based on when we collected the sample after 10 days from the hospital or the quarantine center. And then we took 40 unmatched controls. So what we did, first we looked into the antibody titers, because that was the first thing, why they are not contracting the disease. What is happening? So if you look here, this is basically, we looked into total COVID-19 antibody levels. This is basically from a kit that recently is developed by a Delhi company, where they identify all the antibodies as a total antibody. They call it total COVID-19 antibody. So if you look at here, you see that in context, you have much higher titer of these total COVID-19 antibodies as compared to infected, and controls are definitely, they will be nearly negative. When you look at IgA and IgM, you see that IgA levels are really, really high in these contexts as compared to infected and control cases. This is highly significant. And IgM also showed a trend, but definitely IgA was found to be very high. And we know that IgA, in case of influenza or any flu viruses or any respiratory virus, is highly protective, because this is present in our mucous tract or respiratory tract. And if the specific antibodies are high, then it protects from the infection. Then we looked into the asymptomatic versus symptomatic. What is happening? We definitely see that symptomatic cases had higher antibody titers. But if you look at the significance level, we don't see that they were significant in terms of total cases. They were not 0.05 or something, but they were nearby significant. But we don't see significant difference in asymptomatic versus symptomatic cases. The trend is definitely high in case of symptomatic cases. But these symptomatic cases, I would like to put a disclaimer here that these symptomatic cases are definitely not severe or like from ICU or something. These are mild to moderate cases and not severe cases, because last time we know that a lot of cases were not severe. Now what we did, we used the serum from these individuals and looked into the nucleation as well, using the pseudovirus, pseudovirus nucleation assay, because that you don't need to do in BSL-3 and all. So here you can see clearly that these antibodies definitely in context have protective response also. So contacts have higher like a significant protection as compared to symptomatic, and symptomatic as compared to asymptomatic. So we definitely see that the antibody levels correlate strongly or corroborate strongly with the nucleation activity. And this suggests that that's how they are protected from the virus infection, and they were not coming the qPCR positive. I just added this slide here because we recently started looking into what is the antibody titers in Covaxin vaccinated individuals, because I'm collecting a cohort of Covaxin vaccinated and Covishield vaccinated individuals. So look into it in a longitudinal time frame, what is happening with the antibody levels and as well as the cellular immunity that we have not checked there. So if you look here at 28 days, you will see a lot of individuals, they don't develop antibody. And I'm not showing here graph, but around 90 to 95% individuals after first injection, they don't develop antibodies. But After the booster dose, this is basically 10 to 15 days or 10 days around after the second dose, you see that in all the individuals except 3 to 4%, the antibody levels are really high. After 90 days, you see that the antibody levels are more or less same. In some cases, it went down. And in 20-25 cases, you see that rise is really high. But there are some cases in this also that they got reinfected after vaccination, and in those the titers are really very high. It's like 40,000 or 45,000 units. So this is just, I wanted to show you that what is happening in case of vaccinations. So after that, after looking at the antibody responses, we wanted to look into what is happening to the cytokines. Is there like a signature of cytokine versus antibodies? Cellular immunity is also taking part. So we did this 48 plex, but I'm not showing you the here whole profile. But if you look at these 4 cytokines, this is like cytokines and chemokines, IL-7, MIP, MIF1 alpha, MIF, and GCSF. You clearly see that in context, these cytokines are significantly higher as compared to these patients. These two are basically not present even and more like control in case of asymptomatic or infected cases, but controls, they were significantly high. So this is just a clustering analysis because we wanted to identify, do we see some signature, protective signature of high antibody along with the cytokine signature? Do we see that? So we just did this clustering of these cytokines along with the antibodies. And these are the different controls, asymptomatic, symptomatic, and control cases as indicated by these colors. So this color here, you can see this blue color. This is basically the contacts, direct contacts. You can see here they have really high antibodies, and they show a clustering of these cytokines very strongly as compared to the COVID patients, which are on the left side, you can see here. But they don't cluster into asymptomatic, symptomatic. This is more like a random picture. So it appears definitely there is some cytokine signature along with high antibodies. That response is generated in these direct contacts that is protecting them from contracting the disease. Then we looked into the ROC curve and AUC. What are the cytokines which are showing 80% AUC area under curve? And we see that these cytokines, like 5-6 cytokines, they show that they appear to have more protective responses in these direct contacts. So now, why that is happening? Because now it's fine that these are there, antibodies are there, and then you have cytokine signature. But what is the response by these B and T cells? Because they are ultimately leading to generation of antibodies, they are leading to the generation of these cytokine responses. So do we see a specific signature of B and T cells in these direct contacts as compared to the controls or infected cases? So what we did, we sorted B and T cells from PBMCs of contacts, but we took only 3 contacts, 3 infected cases, and 3 controls. Then what we did here, we use 5' this gene expression analysis along with BCR and TCR, because that is the format that 10x supports. But before going into it, I would like to just tell you about like Devzani and then Nitya. A lot of people already told you about why single cell analysis would be important to do that. As I told you that there are B and T cells that are antigen specific, that will only go clonal expansion. But you will be having a lot of background cells, like as you can see here, the gems. So if you say that red cells are basically the ones that are basically recognizing the antigen, you have a lot of other cells that are present there as well. So that's why if you do a bulk level analysis, you cannot identify what type of gene expression in these red cells as compared to the other cells, and that will give you a very big background. So that's why in such cases, single cell analysis definitely helps, and you want to identify at single cell level the T cell receptor and B cell receptor profiles. Then only you can identify what are the antigen specific T cells and B cells. Why single cell? Again, this is also one of the simple slide I'm showing here. Because if you look at this left side, you have one transcript or one molecule in each cell. Okay. And you count it as 7 when you do bulk analysis. But if you have only 2 or 3 cells that are expressing all these transcripts, others are not expressing, still you will count them as 7. But in this case, this is not true. And this is the biological system where there is always a heterogeneity. Only few cells express very high levels, some cells express medium levels and low levels. And that leads to this heterogeneity. So in real sense, it's very important to understand these cells which are expressing versus not expressing separately to understand what is happening. So I added one more this slide because I felt this slide is definitely explaining what things can be done if you are doing single cells. Because in single cells, as I said just before, that you can deconvolute heterogeneous cell population like B cells or T cells, where you have a mixture of clonally expanded cells versus the other negative population. You can deconvolute and identify the individual population, as I will show you here as well. Then the other thing is, if you want to look into cell state transition, as in case of stem cells and all, you can do trajectory analysis where you can really identify. This is very interesting in case of development of chemoresistance as well, because if you have time dependent chemoresistance in any clinical sample or in cell line, you can try to identify why that chemoresistance is induced in the cells in a time dependent manner if you will do single cell analysis in them. The other thing that is I feel quite interesting is that we have started looking into this dissecting the transcription mechanisms, because you know that when virus or any antigen activate your antigen presenting cell, all the cells don't respond to the same level. As you can see here, these red cells, they are basically heavily transcribing, the other cells are not transcribing. So it is very important to understand why some cells respond, why they generate responses, whereas other cells are not generating responses or they're generating responses at very mild level. So what is the mechanism behind that to make on and off situation? And these things can be also done if you are doing a single cell analysis. So coming to the experimental outline of this study. So as I already showed you the antibody profiles and the cytokine profile. So what we do fact sorting first for the B and T cells from the PBMCs for contact control and these patients. And then we do 10x gem preparation. I'll tell you one more thing here, because when you have isolated B and T cells, you have removed a lot of background if you are studying only B and T cells, because you know that 70% of the cells are T cells and B cells percentage is quite low. So that's why what we did, we sorted here B and T cells, and it's possible to pool 2 or 3 samples in one by hash tagging. So what we did hash tagging here, and we pooled 3 samples in one well of the chip for single cell this what you call separation. And then we looked at the gene expression using 5' gene expression and the TCR and BCR repertoires in these B and T cells, sorted B and T cells. So here I'm just showing the t-SNE plot or the UMAP plot, where you can see these cells in different clusters or colors based on these control, contact, and patients. And here you see that these clusters based on different cell types, T cells, NKT cells, and B cells. There are some monocytes and platelets also, they come as like background cells. So the number is quite less in that case. So first I would tell you about the T cells, what is happening when we did the single cell profiling 5' gene expression for T cells and the TCR repertoire. So we took all cells, but I will what after as I showed here, from these clusters, you can take out T cells to do the detailed analysis, or you can take out the B cells and then you can do the detailed analysis on that. So first I would show you about the T cells and what are the TCR repertoires in these cells taken from contacts, from the infected patients, and the controls. So this is just the UMAP plot showing all the different type of T cells with different colors, because based on the signature of here in the right side, you can see this dot plot. Based on the expression of different genes, signature genes, and different cell types, you can identify these clusters and define that these are the clusters. And then you can look in detail into these specific cell types. What is happening? What are their gene expression at single cell level? What are the T cell receptors, clonotypes that are there? So this is just showing that identifying different cell types, T cell types, in this all T cell population by their gene signatures, the genes that are expressed, signature genes. And this is the cell number that basically total cells were around 6,000 for T cells. This is after removing all doublets, triplets, dead cells, mitochondria, and a lot of filtering criteria. So when we looked into the CD4 cell subtypes or the CD8 T cell subtypes in control, contact, and patient, so what you see here, that in control, you see that there are I would say that directly in context, you see that there is more of CD4 memory T cells as compared to these in patients. Similarly, if you look at this CD4 naive T cells, you have high population. So it appears that in context, somehow the CD4 memory cells are, you see they are expanded memory cells. If you look at CD8 positive subtype, this is only showing the fraction. How many CD4 T cells with different fraction of the CD4 naive, CD4 memory, and Treg based on their uh, gene expression signature. When you looked into CD8 T cells, you see here that these are different subtypes shown as different color. But I would just indicate on the important subtype that we found here that appears to be interesting. So we see that CD8 positive TAMRA cells are somehow very high in fraction in context as compared to the patient, as compared to the controls. And these, I thought that there will be more of cytotoxic T cells, but CD8 TAMRA cells, these are basically CD8 T effector memory cells, which are RA positive, and these cells are reported in inflammatory diseases, autoimmune diseases, to be highly cytotoxic by the granzyme and perforin, as compared to even CD8 only effector memory cells or CD8 effector cells. And they are reported recently in a lot of inflammatory diseases. So we see that CD4 memory T cells are highly enriched, and CD8 TAMRA cells appear to be really high clonally expanded in context as compared to the patients. This is just showing the gene expression, 5' gene expression for the TAMRA cells. You can look at cells, but we focused here more on TAMRA cells for this work. And then we found that these CD8 TAMRA cells, they have more of antigen processing, presentation of exogenous peptides, genes highly upregulated. Then they have interferon gamma cellular response upregulated. So it appears that these cells, CD8 TAMRA cells that are in context, they are basically somehow showing much more of inflammatory type response or more of cytotoxic response as compared to patients, which might be one of the reasons for clearance of virus and the symptoms. So if you look into now the TCR repertoire of these cells, because first you look at what is the fraction of different T cells. Do you see some specific T cells that are enriched? And then you go for what are the TCRs specifically expressed on these clonally expanded cells. So this is just looking into as a general, how many TCRs are expanded on how many cells, T cells in this. So these colors represent basically the hyper expanded TCRs, large expanded. So these are the number of TCR clonotypes that are observed in these different cell types in control, contact, and patients. Okay, and these are the TCR percent TCR present on CD4 naive, CD4 memory, because these are all T cells. So you look into how much percent of TCR you found on different T cell types. Now, overall, if you look into the TCRs in control, contact, and patient, you see that in controls, you have less expanded these clonotypes as compared to somehow we see this hyper expanded clonotypes, and this we are still looking into why this control is showing. Is the control having some asthmatic or some allergic response which lead to already some T cells which are hyper expanded in one of the control? If you look into the context, you see here the major portion shows that large, medium, or small expanded clonotypes as compared to patients. In patients, you see that the TCR clonotypes are not that expanded, and they have single like clones in a lot of uh, CD4. This is for Tamra. So you have more of the single TCR as compared to the clonally expanded TCRs. If you look here at the same way and different cell types, so I will not go into all the details, but if you look at here CD8 Tamra, you clearly see that you have highly expressed this like TCRs is medium expanded and large expanded, is basically saying that one or two TCRs are highly expanded and those cells are clonally expanded, basically leading to the clearance of the pathogen or infected cells. Now, if you do the clustering, this is much more easy visualization to show that which cells are basically leading to this increased clonotype. So if you look here, these are basically based on color. These are controls, contact, and these are patient samples. So, and this line, pink line here, this shows that which clonotypes in which sample, because this yellow is the contact. You see that the thick, this pink line is going to the CD8 Tamra here, and CD8 Tamra TCRs are highly expanded, and this is the clonotype written here. We know even the number of copies of these clonotype in these cells, how many number of copies are there. Whereas there are some clonotypes which are very less. You can see here they're thin pink lines in different cell types. But you see strongly CD8 Tamra and this specific TCR subtype, TCR alpha, TCR beta subtype of TCRs are clonally expanded in CD8 Tamra effector cells in contexts. But if you look at the patients, what is happening? In patients, you see that you don't have any TCR clonotypes highly expanded on TAMRA cells, but you see that there are NK cells where you see this NK or this, yeah, definitely NK and NKT cells, where you see that hyperexpansion of this clonotype. And this is the clonotype that we identified in this patients. So there is a difference between contacts and patients in terms of the TCR clonotypes that are expanded. So this is quite interesting that we see. That is the beauty of the single cell, that you can really go stepwise looking into different clusters, and then what is really happening in terms of gene expression, in terms of TCR or BCR clonotypes. What is happening to the B cells and BCR clonotypes? So if you see here, now these are the clusters. So what you do basically in the initial t-SNE analysis, you first identify all the B cells, T cells, and all these things, and then you take out the B cells and you look into the different subtypes of B cells inside. So here, on the basis of this gene expression signatures, you differentiate them into naive B cells, class-switched memory B cells, unswitched memory B cells, and plasma cells. Plasma cells somehow we missed in this because we use CD3 and CD19 for the sorting, and plasma cells have CD23 or some other signature that we didn't use. This is only the neglected, or what you call the inflate background cells that are coming during the sorting. So we can focus only on naive, class switched, and unswitched. So what is happening exactly in this, if you look at the fraction in control, contact, and patient? So you see that in control, you have less of class-switched memory B cells, which is expected because they have not seen the pathogen. But in case of contact and patient, you see more or less similar type of class-switched memory B cells. There is not big difference. There are some plasma cells in patients. During sorting they came, so we don't need to focus on that. Even if you look at the individual control and patient and contact samples, so these two are controls, these three are contexts, these three are patients. So this is somehow the ID is left here. So we see that in contact and patient, we don't see big differences. In terms of proportion, it looks more or less similar. But if you look at the clonotypes, you see that somehow in context, you see a little bit more clonotypes as compared to patients. In controls, you don't see these expanded clonotypes in BCR, B cell receptor. If you look at the IgG clonotypes, IgSD, IgG, different type of IgGs, you see here that in case of contacts, you see that this IgHG clonotype is expanded. But definitely in case of B cells, we don't see very like stark differences between contact and patient. Our idea was more to look into contact versus patients, as I told you, because the idea was to look into why contacts, direct contacts are not infected in contracting disease. Control, we took it just for the comparison to see that same PCR, same BCRs are not coming. Now, if we look into the VDJ recombination, because this is also a group, but if you look at how the V and G genes are recombining in controls at a single cell level, at controls, asymptomatic, symptomatic cases, and contacts. So if you see that these signatures look quite similar in controls, asymptomatic and symptomatic, but here if you can see that IGHV 333 and J4, the signature is reversed here. So you have more of 33 and J4 in case of controls, which is reduced, and you have more of 323 and IGH J4 in case of patients, which is similar in context. But in context, you see some more recombination happening, and you have increased copy number of these ones, this 214 and J2, and then this one. So definitely there is more of clonal expansion happening in context just after short exposure, or they have already seen that similar type of pathogen with antigenic peptide. So they already have some memory B cells and T cells that underwent expansion immediately. They have high IgA. They have this protective cytokine signature that lead to protection, most probably lead to protection from contracting the infection and the symptoms. So these are the observation and conclusion from the study. So direct contacts showed definitely higher titers of virus-specific antibodies, specifically IgA, which are present in the mucus. High antibody titers in contacts correlate strongly with high virus neutralization capacity. So yeah, this is just the last slide. So so basically these cytokines, 4 or 5 cytokines, show really strong, more than 80% AUC. So it appears to be more protective. Context depicted increased class-wise memory B cells and CD8 T TAMRA cells. The CD8 TAMRA cells and their clonotypes that we found, that is very, very interesting part that we identified here. So it appears that TCR clonotypes with viral recombination events are enriched in contexts as compared to the cases and controls, and that is basically enhancing the cellular immunity. So at last, I would like to acknowledge, because this was the work that is done during really difficult times, because there were lockdowns and a lot of things. So here, Kaushik, and up to some extent, my very senior PhD student, Abdul, he helped him in the initially setting up the single cell experiments. He is now in National Cancer Institute. And Kaushik did all the experiments for sorting and everything. And Arup initially set up the pipelines for analysis of single cell data and all. But now Sudeshna is doing all detailed analysis for single cells and all clustering and everything. So she is now expert in doing this analysis. And this is my group, different people. Last year we took this with mask and all, so still we are wearing it. And at last the funding agencies, because before, without that, it was not possible. So DST, DBT and all, ILS for all the support. And one more thing, we have COVID like this sample repository. I would like to acknowledge that, because we took the samples from them and the core facilities of ILS, without which it was not possible. Thank you. Thank you. I would looking forward for the questions then. Yeah. I hope it's within time.
Yes, sir.
I can remove this now, maybe.
Thank you, Sunil sir, for a wonderful talk. We are having a separate discussion time after all the talks for the day. So maybe we can take questions at that time.
I just have to remove this now.
You just have to stop sharing. There will be a button on the top. From the top, you will get.
Stop sharing. Okay, it's fine.
So the third speaker for session 2 is Dr. Deepak Modi. He has done his PhD in human genetics. He is recently elected as a fellow of National Academy of Sciences India 2020. Presently, he is Scientist F at the ICMR National Institute for Research in Reproductive Health and also heads the Molecular and Cellular Biology Group. He has over 85 publications in several prestigious national and international journals. His current research areas include understanding of endometrial receptivity and endometrial disorders, genetics of sexual development and male fertility, COVID-19 and reproduction. He is also the co-investigator of the Pre-COVID registry, which collects information on pregnant women with COVID-19 and their babies in India. The floor is all yours, sir. Thank you.
Thank you, Mamta. I hope I'm audible.
Yeah.
Slides visible?
No, yes, sir.
Just a Thank you so much, Mamta, and thank you, Sunil, for alluding us to the wonderful work that you have been doing at ILS. I'm really awed with the work that has been discussed in the day since morning by Ankur as well as you. And we have done something in single-cell RNA-seq, but in a very, very frugal manner. And what I would give you an overview is what something that we have achieved in the last 1 year on how single-cell RNA-seq analysis has helped us to understand the maternal to fetal transmission of SARS-CoV-2. As Mamta said, that our group is basically interested in understanding initiation of pregnancy, early placentation, and disorders of sex development. I have a wonderful group of students, postdocs, and scientists who have help us to do a lot of things. But specifically, all the data that I'll share today has been the large work contributed by Nancy, Tomda, Anshu. Karishma was a Fulbright Fellow, and Stacy and I gratefully acknowledge their hard work in the difficult times, working from home and remotely working throughout trying to get a lot of information. So when it comes to infections, one of the problems is that when a pregnant woman gets infected, one of the basic risks is the transmission of the infectious organism from the mother to the child. Not all organisms get transmitted, but if at all there is a maternal infection, there is a likelihood that there could be a transmission during pregnancy, during labor or delivery, and during breastfeeding. So the transmission during pregnancy, if there is a congenital transmission, then it can have ill effects on the baby like abortions, stillbirths, congenital malformations, and growth retardation, the Zika virus being the classical example. And if there is a transmission during labor and delivery, it can lead to preterm birth, rupture of meninges, septicemia, and breastfeeding infections can lead to newborn. Now, when it comes to SARS-CoV-2, earlier it was thought a disease of the old, but now we know that a lot of young population is getting infected. People in reproductive ages are getting infected, and pregnant women are also getting infected. So the question at the beginning of the pandemic that we asked was that what is the impact of SARS-CoV-2 in pregnancy? Do pregnant women can have any negative impact from this? And towards that, my colleague, Dr. Rahul Kajpi, has started the Pre-COVID registry. And in parallel, our lab started asking whether SARS-CoV-2 can be vertically transmitted from the mother to the child. The talk is divided into two aspects. We wanted to see if SARS-CoV-2 can infect the early embryos, that is even before the pregnancy is initiated and the embryos are there. Are these embryos susceptible to SARS-CoV-2 infection? Or is the developing placenta susceptible to SARS-CoV-2 infection? I'll tell you in a minute why we will placenta. The question was that are these embryos or placental tissues or fetal tissues likely to get infected because of SARS-CoV-2? To address this, we took a very straightforward, simple approach. We said that, okay, if the SARS-CoV-2 is to infect the cell should have the necessary machinery for infection. When we started this work in March last year, we already knew that SARS-CoV-2 utilizes the receptor ACE2, on which the S protein binds. The TMPRSS2 is the enzyme which cleaves the S protein, and this helps in viral endocytosis. We now know that this is a canonical receptor, but there are several non-canonical receptors like Basagin, NRP, Golga7. And along with TMPRSS2, there are these non-canonical proteases like the endosomal proteases, cathepsins, which help the virus entry and its further pathogenesis. Once the virus enters inside the cell, the RNA is processed, the viral proteins are made into the Golgi complex, and then the viral replication occurs, and the new virus particles are set in, and the virus is then thrown out. Now, this entire process from entry to the exit utilizes a plethora of host proteins which are there inside the cells, and absence of some of these proteins can reduce viral infectivity, and the presence of some of these proteins can increase the susceptibility of the cells for the infection. So we asked a straightforward question that are these proteins or are these RNA present in the human embryos or the placental cells? Now, classically, one would actually take these tissues, do RT-PCRs, immunohistochemistries, Western blottings to do this, but since the labs were locked and we would not have access to these tissues so immediately, we thought, okay, let's look at publicly available database. Now, the good part of single-cell RNA-seq is all the work that's been done by a lot of people are actually deposited, and you can have access to it. So we thought that why not make use of this rich source of information which is there? Let us mine this information to see if there are any SARS-CoV-2 receptor expressing cells in the developing embryos. The good part for us was there were a couple of data sets already available. Yan et al. Just the year before it published a data set of about a good number of embryos, or good numbers of cells from about 100 plus embryos, which were obtained from IVF. Even remember, these are human embryos obtained at IVF. Every single cell of these embryos was sequenced, and the data was there. They'd used it for a completely different purpose. We took another data set from Petropolis, where they had sequenced about a large number of blastocysts from day 5, day 6, and day 7. There were a large number of cells sequenced. So we thought, let's look at these two data sets and see if the necessary receptors for SARS-CoV-2 infection is there in these cells. And here is a bubble plot. Here you see ACE2 from the developing zygote, 4-cell, 8-cell, 16-cell. The epiblast and the primitive endoderm are the cells which are in the inner cell mass, which is going to form the body. And the blue cells here are the cells of trophiectoderm, which is going to eventually form the placenta. These are pre-implantation, implantation stage embryos. And here we could see that there are definitely ACE2 expressing cells. The ACE2 mRNA was detected in the zygote, 4 cells, inner cell mass, epiblast, primitive endoderm, and also the trophiectoderm. This was surprising for us. We never knew that ACE2 would do something in these embryos. We never thought it would be there, but we could actually find it. And corollary to that, we did not find TMPRSS2 and all the cells, but the epiblast cells, which are a small subset of cells with ACE2, also express TMPRSS2. And most of the trophiectoderm cells also express TMPRSS2. So these results told us that the canonical ACE2 and the TMPRSS2 mechanism is expressed by the human embryos. We also found that the non-canonical receptors like Basagin and NRP1, Golga are also expressed by the developing embryos. We also found the CTSLs, the non-canonical mechanisms of SARS-CoV-2 entry material was also present in the human embryos. Even the receptors for other coronaviruses like the MERS and the nCoVs were expressed by the developing human embryos. Here we show the T-sign plots. And we could see here that these purple colored cells here are the epiblast cells. And as I told you, a subset of epiblast cells express ACE2 and the same cells express TMPRSS2. Zygote cells did express ACE2 but did not express TMPRSS2, indicating that they may not be directly related to SARS-CoV-2 entry. And this is the trophectodermal cells from Petropolosis data set. And we found that the orange color, which are the day 5 and the day 6 blastocysts, almost every cell expressed ACE2 and TMPRSS2, whereas the day 7 blastocysts, 40% of cells express ACE2 at lower levels, but they also co-express TMPRSS2. We went one step ahead and we said that, okay, the receptors may be there, but what if the machinery for endocytosis is absent or the machinery for replication is absent, then the infectivity may not be very high. But contrary to our expectation, we found that there were about 45 genes which are known to be required for viral replication. And cells from the compact morula onwards and the trophectoderm, almost 50 to 60% of these genes were all expressed at reasonable levels in the developing embryos, right from compact morula to the trophectodermal stage. Also, at the same time, the viral replication genes, these are genes used by the present in the host, used by the virus to increase the replication, and a proportion of them were present. When we were analyzing this data, somebody published a data set where they identified about 333 host proteins which physically interact with SARS-CoV-2. And we found that out of these 333, nearly 60% of these host proteins were expressed by the trophectodermal cells. Eventually, of this 333 host proteins, 63 proteins are absolute necessary for viral infectivity. So if you reduce these proteins from these cells, the infectivity goes down. And so we call them as coronavirus infectivity-related genes. And all these infectivity-related genes were also found in the cells of this early developing embryos. And that made this figure to summarize the results, and which told us that the cells from the early zygote to the morula stage do not have much of receptors. They have low abundance of these infectivity-related genes. But as soon as the embryo becomes the blastocyst stage, when it is ready to implant, there is expression of ACE2 and TMPRSS2 on the trophectodermal cells and on the some cells of the inner cell mass. Now, this has a lot of implications clinically. Now, IVF clinics, women who are undergoing infertility treatment and they have the embryos in the lab petri dish, the labs have to be careful because this is an asymptomatic disease in many individuals. Asymptomatic individuals can spread the virus, and if this virus gets into the IVF clinics, into the laboratories, the embryos can potentially get infected. And a paper has come in bioarchives very recently, which did show that pseudoviruses, when embryos are incubated with pseudoviruses, the embryos undergo a lot of damage and many of the embryos die, indicating that SARS-CoV-2 could be embryotoxic. And this could mean that if individuals are infected, or trying to conceive and get infected, they could have miscarriages, or in IVF labs, and the embryos are being cultured, and if there is an infection there could be adverse effects on the embryo. I'll switch gears now that the woman is pregnant. Suppose there is a pregnant woman who is getting infected, and this is one of the most dreadful conditions because here we have no control. At least in IVF labs, have some controls. If the pregnant woman is getting infected, can she pass on this virus to her baby? Now, not all infections can get passed on from the mother to the child simply because the placenta acts as a nice barrier. Now, if we hypothesize that if the placental cells do not have the necessary machinery for SARS-CoV-2 infection, the virus cannot cross the placental barrier and therefore it cannot infect the baby. Or corollary, if placenta has the necessary machinery for viral infection, then there is a possibility of the vertical transmission or mother-to-child transmission of the virus. So we again took the same approach. I'll just quickly run you through the placental cells that are there in the normal placental villi. The outermost layer are the syncytotrophoblast cells. The inner layer, which is the dark pink here, are the cytotrophoblast cells. And the big cluster of cells which you see here are the extravillous trophoblast cells. And the virus can, from the mother's lung, it can get into the bloodstream. In some of the cases, it can come into the bloodstream. The maternal blood is floating all around here. And if the virus is there in the bloodstream, these cells potentially can get infected with virus if they have ACE2 and TMPRSS2. We again use the same approach. We took two additional data sets, which are of the first trimester human placenta and the second trimester human placenta. Remember, we did not do any single cell RNA sequencing ourselves, but somebody had done it, and this data set was already available on the World Wide Web. We downloaded it and started analyzing for the presence of S proteins in the trophic-terminal cells. And here is the data. You can see STP as syncytotrophoblast cells. A number of STPs did have ACE2. A small but a significant numbers of cytotrophoblast had ACE2. A small number of EVTs also had ACE2, and the stromal cells, which are the cells of the villus core, also had ACE2 expression. When we looked at TMPRSS2, unlike the trophectoderm of the blastocysts, TMPRSS2 was not abundant, but there were a proportion of cells which also did express TMPRSS2. The alternate receptors, basogen and the capsin L, abundantly almost every cell of the placenta had basogen, had CSTO. And when we looked at the first trimester over the second trimester placenta, we found that in the extravillous trophoblast, which are going to do spiral artery remodeling, there was in fact increase in ACE2 expression during in the second trimester. Even the TMPRSS2 expression significantly increased. Basogen also increased and CST also increased. So these results together told us that possibly the placenta also may have the necessary machinery for SARS-CoV-2 infection. These are UMAP plots. This was done by Monique Charlie at ISC, who was a collaborator with us with this project. And we could show very nicely that the ACE2 and the TMPRSS2, there were ACE2 and TMPRSS2 co-expressing cells, specifically in the second trimester extravillous trophoblast and the first trimester syncytotrophoblast, as well as some of the cytotrophoblast cells. Now, as in the morning, we were discussing that, okay, this is RNA, does this form the protein? So eventually we could also do immunofluorescence for SARS-CoV-2 in the first trimester placenta. And corroborating our single-cell RNA-seq data, we could see that there were syncytotrophoblast and cytotrophoblast cells nicely expressing ACE2. You can see this red staining here, and there are not every cytotrophoblast cells express ACE2. You can see a subset of cells expressing ACE2 in the placental villi, indicating that yes, there is mRNA and the protein was made, and therefore if this protein is there, if it experiences a virus, the SARS-CoV-2 would internalize. Of course, we did a whole lot of experiments over that, but I'm not going to take you to the details of this, but we wanted to characterize what are these syncytotrophoblasts which are co-expressing ACE2 and TMPRSS2. We looked at all the gene signatures of the ACE2 TMPRSS2 negative syncytotrophoblast and ACE2 TMPRSS2 positive syncytotrophoblast, and to our surprise, we found that the gene expression signatures were very different, indicating that the metabolic statuses of the cells are highly divergent. And we found that these cells were enriched for pathways involved in monosaccharide transport, adherent junction assembly, actin polymerization, depolymerization, cell proliferation, and an interferon signaling pathway. And based on the marker gene analysis, we could show that these syncytotrophoblasts, which are expressing ACE2 TMPRSS2, are of a highly differentiated variety, which are supposed to be doing all the transport of nutrients from the mother to the child. That means the cells which are very crucial for nutrient transport from mother to the child are positive for SARS-CoV-2 and for SARS-CoV-2 receptors, ACE2 TMPRSS2, and therefore they are likely to get infected. And we could further show that these cells which are ACE2 TMPRSS2 high also have abundant expression of genes required for viral endocytosis, viral replication, and SARS-CoV-2 protein-protein interactions and infectivity-related genes. We also looked at the extravillous trophoblast cell population. What are these cells? Now, I forgot to tell you that these extravillous trophoblast cells during pregnancy have to do spiral artery remodeling so that the placenta anchors itself and there is an easy exchange of nutrients from the mother to the child. What we found was that unlike the ACE2 TMPRSS2 cells, the gene signatures of the extravillous trophoblast was quite noisy there, but still from this data, we could identify that these extravillous trophoblast cells, which are ACE2 TMPRSS2 positive, have abundant expression of genes required for viral release, genes required for viral entry, indicating that they're quite susceptible to viral entry. And there were genes involved in epithelial migration and wound healing, indicating that they could be migrating cells. Now, of course, extravillous trophoblast cells, the job is to migrate, but based on marker gene analysis, we found that these are the extravillous trophoblasts, which are left the trophoblast column, and these are the migrating trophoblasts, which are supposed to line the spiral arteries, have ACE2 TMPRSS2. And also, like the syncytotrophoblasts, they have abundant expression of these genes involved for viral endocytosis, replication, and SARS-CoV-2 interactions. So this work got published in Frontiers in Cell and Developmental Biology in August last year. And we could show that almost multiple cell types of the developing placenta have expression of SARS-CoV-2 receptors, with highest being in the syncytial trophoblast and the endovascular extravillous trophoblast at the maximum number of cells with SARS-CoV-2. Now, when we published this, of course, a lot of people asked that, okay, fine, these receptors are there, but does the virus ever reach the placenta and infect it, or it is just an imaginary situation? I'll just quickly take you through a case report which we came across at that point of time. We thank Dr. Prajakta Shinde and a team at ESIS Hospital and Dr. Rahul Kajbir, who managed to get these samples for us, and our Dr. Vaina Patel and Vikrant Kaur for helping us to do the SARS-CoV-2 analysis. So this was a woman who was infected at 8 weeks of pregnancy. Very early in pregnancy, she had an ultrasound done and the fetus was good, but she got infected with SARS-CoV-2 and she had a completely asymptomatic course, and her CT values were also quite high at 28, indicating it's a mild to moderate infection. And at 13 weeks of gestation, she came back to the hospital for a routine ultrasound checkup. And what did we find? The doctors found that the fetus had all the fluid accumulated around it, which is called as hydrops fetalis, and there was no cardiac activity, indicating that there was an intrauterine fetal demise. The doctors called up and asked whether this could be because of SARS-CoV-2 infection. We were not sure because the patient was infected almost 5 weeks ago, mild, completely asymptomatic course. And at that point of time, we didn't know whether asymptomatic infections could really do anything bad. We thought, okay, there is no cytokine storm, so nothing really should happen bad to the fetus. But we said, okay, let's investigate that. So when she underwent abortion, her swab was negative for SARS-CoV-2, indicating that she had cleared the infection from her throat. But when we did the placental tissue and did the RT-PCR, we did find the presence of the viral RNA. Amniotic fluid, which was collected from the fetal sac, was also positive for viral RNA, indicating that the virus still persisted almost 5 weeks after the primary infection. This was surprising for us. So we call this as persistent placental viremia. So the maternal system cleared the virus, but the virus is still there in the placenta. We were asked that whether this could be just simply that the maternal blood is having the virus and the placental cells are not really infected. That was not the case. We used immunofluorescence here. Anshu came in the middle of the lockdown and we did a whole lot of experiments to get this thing up and running. Here is S1 and S2 protein of the virus, the spike protein of the virus. And ladies and gentlemen, here you can see the virus actually happily sitting in the villus of the placental cells. Remember, the mother's throat swab is negative. She does not have the virus in the throat swab, but the virus is thriving in the villus cells. You can see the syncytial cytotrophoblast nicely expressing S1 and S2 proteins. And here is a zoomed image, which is another viral protein, non-nuclear capsid protein, which was localized to the syncytial trophoblast along with ACE2. Remember, I told you that ACE2 positive cells should have SARS-CoV-2, and yes, we did find co-localization of SARS-CoV-2 virus proteins in the cells which are ACE2 positive. We also found some cells which are ACE2 negative but still have the virus, indicating that there is a possibility that also through the non-canonical route, the virus can enter inside the cells. This work got published in Human Reproduction late last year, early this year. What we could also find was, using the single-cell RNA-seq data, that this ACE2 TMPRSS2 positive syncytial trophoblast and the extravillous cells which are likely to get infected with SARS-CoV-2, and we could show that they do get infected with SARS-CoV-2. They have abundant expression of toll-like receptor signaling pathway, apoptosis signaling pathway, and JAK-STAT signaling pathway. And we thought that this could mean that there could be inflammation happening there. And yes, in this patient where there was SARS-CoV-2 infection in the placenta, we could see a whole lot of immune cells in the intravillous spaces. Normally, you would not see immune cells in these regions. You would see these are devoid of immune cells, but we could see there was a lot of immune cell infiltration in the intervillous space and also in the decidua. There is regions of decidua which were filled with inflammatory cells sitting there. That means that the virus not only infected there, it replicated also around and mounted an immune response. And we think that this entire mixture could have led to fetal hydrops and ultimately fetal demise. So this is how we now propose that there could be a mechanism. So using the single cell RNA-seq data set, we could now show that if there is maternal infection of the virus and if there is a systemic viremia, the virus through the maternal blood can reach the placenta. In the placenta, a subset of cytotrophoblast and cytotrophoblast cells have the ability to get infected. These cells have the necessary machinery for viral replication. So the virus can happily stay there and replicate it and increase in the numbers, causing placental infection. And in the placenta, you can see, we did show the infection here. And then once a placental infection happens, and the virus can easily seep into the fecal circulation, go up to the fetus, get to the amniotic fluid, go there, and then infect the baby, leading to a mother to child transmission. So this is a first case where we could show that a first trimester infection of SARS-CoV with the mother could be detrimental to the pregnancy. So to summarize, using the power of single cell analysis, we could show that the early embryos, even before they are ready to become pregnant, the early embryos are permissible to coronavirus infection. In the placenta, multiple cell types are permissible to SARS-CoV-2, and SARS-CoV-2 can actually infect the placental cells, replicate them, and this could lead to vertical transmission and inflammation. This is where I'll stop now. I'm grateful to my staff and students and to Dr. Rahul Gajvi, Dr. Prajakta Shinde, and Dr. Mohit Jolly for helping us to do this work. And funding is from ICMR intramural grants. And my lab is also funded by TBT and DSD and Influencer. Thank you, and I'll be happy to take questions.
Thank you all the three speakers for such a great talk. Now we will begin with the discussion session. The first question is for Dr. Deepak Modi. The question is there an increased mortality rate during pregnancy? If so, do we know exactly why that is the case? And it is something specific to the strain currently prevalent in India.
Yes, using the pre-COVID registry data, we are currently analyzing it. Dr. Rahul would be the appropriate person to answer this question, but on his behalf, I'll take the liberty to do that. We have analyzed data of the Maharashtra population, pregnant women population within the Maharashtra belt using the pre-COVID registry data, which is collecting information from 19 medical colleges in the state. And we do find that, yes, pregnant women are not at an increased risk of infection. They're not at an increased risk of symptomatic infections, but those who get infected are more likely to get a severe infection, severe hospitalization, and there is a higher risk of death amongst those infected. Two, women, at least in the first trimester, seem to be at a slightly higher risk of undergoing abortion as compared to infections in the third trimester. I hope that question is answered.
There's another question for you, sir. The dynamic of immune modulation at feto-maternal interface may be influencing the susceptibility of the fetus to COVID-19 infection. So can you just shed some light on the same?
Would you repeat the question? Sorry, the dynamics of?
Immune modulation at feto-maternal interface can influence the infectivity.
Okay, so yes, it does. Currently, we do not have data on what are the immunomodulators happening in the placenta of the COVID-infected and uninfected women. There was just paper which has just come last week, which does show that there is a lot of inflammatory response in the placenta of women infected with COVID-19, whether the placental cells are there with the virus or not there. We are currently doing a project with Varia Hospital in the neighboring hospital with Dr. Ketri Kulkarni, where we are doing a case control study of women with and without infection. We'll now look at the immune cell repertoire of the placental tissue to see if that's the situation. So at present, we don't have much information.
Okay. The next question is for Dr. Sunil Raga. I hope you're here, sir. Dr. Sunil Raga?
Yeah, I'm here.
Yeah. So the question is for you. The Covaxin contain entire virus because it is a heat inactivated vaccine. How would you measure the antibody titers in the vaccinated people and confirm that antibodies are specific for the vaccine?
Oh, it's specific for the vaccine. That is difficult to say if it's specific for the vaccine, because generally it's checked for the specific epitopes, and this vaccine is generated against Wuhan strain. That was not like a mutated strain for spike epitopes and all. So I don't think it will it should recognize the same way as the uh, vaccine, like the antibodies are recognizing the RBD domain in the kits. The only problem is if you say for these new variants, if you want to say for the new variants, if those antibodies are really responsible or not, that is still a question. That a lot of people are looking into now. If they are utilizing the new virus strains like 617.2 and .3, we don't know. That need to be looked into. But the
Okay.
antibody here identified in this, that the titer I showed, that is basically for the Wuhan strain, the RBD domain that is used for in the kits for estimating the titer. So it should not be a problem.
Okay. There's another question. Is there any study confirms that Indian variants escape the immune response?
Yeah, there are several studies now. So it's very clear that 617.2 and 617.1, the neutralization capacity for the antibodies that are generated by Covaxin and Covidshield, they are 5 to 7-fold low. So they are not neutralized by these antibodies from Covaxin and Covidshield. So definitely the variants are have immune escape.
Thank you, sir. The next question is for Dr. Devjini. Devjini, ma'am, are you here? Dr. Devjini?
Yes, I'm here. Yes, I'm here.
Yeah. Question for you, ma'am. How effective the genetic panel for cancer diagnosis is, and despite the mutational link which drives genes and passenger genes?
So it totally depends on the problem at hand, and the content that's covered in the gene panel and the depths of sequencing that you're trying to achieve, right? So you have to have a balance between what you're trying to investigate, what is it that the panel covers, and what is it, the depth of sequencing that you're hoping to achieve. Because depending on the allele frequency, at times the mutation is present in a very small allele frequency, in which case then you have to make sure the panel has enough probes to cover the region of interest, and second of all, your sequencing depths are enough to be able to cover those smaller allele frequency. So you need to have the right technology, the right content in terms of the panel, and the means to getting to that end.
There's another question for you. With newer technology, does this mean that we cannot rely on the research that has been done till date with respect to differential expression of genes in bulk samples?
No, not at all. In fact, bulk will give us a broad indication. We're just trying to say that there are additional layers of information that you can get from single cell sequencing that we might miss otherwise through bulk data analysis. We're not saying that bulk data is to be completely disregarded, because that will still give you a broad indication of what's happening. But the additional layers, or at times biology is driven by cells which are relatively lesser in population, which you might miss in bulk data analysis, and that is what we're hoping to get through the technologies like single cell genomics. But that doesn't mean in any way that bulk data is to be disregarded.
Okay, thank you, ma'am. The next question is for Dr. Deepak Modi. So the question is a little big, so I will read it very slowly. So it is known that the cells of the placenta and the embryo express ACE2 and TMPRSS2. Evidence of vertical transmission of SARS-CoV-2 are also documented, mostly in third trimester. Is there a known window where there is a higher co-expression of ACE2 and TMPRSS2 along with the viral infectivity and the replication genes which may be leading to this?
We have done it for the first and the second trimester. The third trimester data was done by somebody else. So it looks like that the third trimester, the co-expression is the lowest, and the first trimester, the co-expression is the highest, indicating that most likely that the vertical transmission and infectivity could be in the first trimester. But since there is no data available in this direction, we cannot really for sure comment.
Okay. This is another question for you, sir, from Dr. Sunil Raghav. Did you check the viremia levels in the pregnant women from where the fetus were analyzed?
Like as we speak, the other project which is being done, we have done the case control studies. We are looking at placental viremia, systemic viremia, and fetal systemic viremia, and we'll bring a correlate of whether that's the case. This particular case report we did not. We never anticipated this at that point of time.
That would be interesting to look into the correlation. Yeah.
So I think we should speak. We need to look at more of immunophenotypes there.
Okay. Yeah.
So the next question is for Dr. Sunil. So how would we know that the circulatory vector or memory cells in the infected patients are specific to COVID antigens? Again, I guess it's the same.
That's very interesting question, I would say, because I think I didn't say exactly that thing. Now what we are going to do is, from the same patients, we have the PBMC distal aliquots. So what we are going to do, we are going to activate in vitro those PBMCs or T cells and B cells with antigens like spike antigens and nucleocapsid. And then again, we will profile those TCRs and BCRs, and then we'll compare. So then you can know that is the same TCRs, BCRs that we identified in TAMRA cells or the other cells, their antigen is specific or not. And that would be very interesting to say that these TCRs are very specific to antigen then, and they are protective. That would be the best thing to do. Yeah.
Okay.
That's
Thanks.
interesting.
That's another question for you. Is it possible that a person do not mount adaptive immune response even after infection with SARS-CoV-2, or we assume that the virus has cleared off by innate immune system?
That's a very big question. So basically, it's really difficult to say, but definitely if the virus load is very mild and innate immune cells have cleared it, it's quite possible, but rare, because adaptive immune response will come up as the dendritic cells or APCs will detect it, and then it will take it. But the only thing is how much cellular immunity is generated. It depends on the viral load and a lot of things. So.
Thank you, sir. The next question is for Dr. Devjini, ma'am. Are you here? Dr. Devjini?
Yes, I'm here.
Yeah. The question is, does pinpointing a particular signature markers to immune response have a risk of underestimating the complexity and the heterogeneity of the immune system?
Not at all. I won't say that. And like I said, there are multiple approaches to actually investigate the molecular mechanisms which are underlying a specific phenomenon that you see. So specifically pinpointing a certain, one certain marker, uh, I would say would add up to the bevy of information that we have. And then it's not like that's going to be the be all and end all, because every study is basically an ongoing process. So I think if at all, it's basically going to enrich the existing bounty of information, and it's going to help us basically focus on specific markers than just hunting around in the dark.
Okay, ma'am. Thank you. The next question is for Dr. Sunil. So if the person is infected second time with the COVID-19, whether the immune response will be different or it will be much faster than the previous infection? So you're not audible.
Yeah, definitely it would be faster, because that's how the adaptive immune system works. So you already have the memory B cells and T cells. it appears definitely that T cell response is much more stronger somehow in SARS-CoV-2 as compared to B cell. And there are other papers also coming that show that T cell response is very strong, T cell memory, and definitely it's much faster.
Okay, thank you, sir. The next question is for Dr. Deepak Modi. So, due to vertical transmission of virus from mother to child, whether child will have any genetic disorder?
We don't know still. There was just this one paper which came which showed that the SARS genome can integrate into the human genome, which is again still a controversial thing. It's difficult to speculate. It's this one case report where we did find those fetal eye drops, which is most commonly known for infections, but we still don't know that it could be. Only long-term studies will tell us.
Okay. There's another question for you, sir. Whether new strain of virus will have a severe effect on a pregnancy?
We don't know. Dr. Rahul is currently analyzing the data. We should have that information in the next couple of months.
Okay. So with this, I guess we have covered almost all the questions. There's one more question from this. Yeah. Dr. Sunil, it's for you. So how did you differentiate between the controls and asymptomatic patients apart from RT-PCR? Whether there was other
That's
tests?
a very interesting question, because how you really know that controls were not infected? But we have a lot of samples basically that we preserve and that were before COVID time. So for controls, we use those samples which were not like during COVID time. So we use the PBMCs that were preserved before, because there are high chances that they will be asymptomatic. Definitely. That's a good question.
Dr. Sunil, one question from my side is, what is the COVID status of your control right now, or the contacts? The contacts, are you monitoring?
It's difficult to follow up because all were migrants. They went back again to Gujarat. So now where they are is very difficult to find out. Tracking longitudinal study with these individuals are very difficult. We thought of, but
okay. Okay, so that data is not available.
No, longitudinal is not there. No.
And any other parameters you captured from the migrants regarding like some food habits or some specific thing that has been captured?
It was even difficult to get the samples.
Okay.
Actually, you must be congratulated for actually getting this work through during that pandemic time, where even going to the lab was impossible.
And I think Dr. Sunil's work is one of the very few on host immune response in the country. I don't think we've had too many on the host immune response.
So this was the first immune experiment of India on COVID, or any first single cell immune experiment.
Yeah, absolutely. So and still is probably
Deepak also did the same thing, single cell immune response. You can't say it's the only one. Yeah, but definitely, there should be more such things happening. And then it should come up from several places. That's very important. We are lacking that, for sure. Now we have good cohort of individuals that we collected PBMCs in a longitudinal way, where they were infected and they had like high titer of antibodies and they went down.
Yeah.
Like, so antibody. So what check like what is the cellular responses in that? Do they have cellular responses, or that is also?
There's one more question for Dr. Deepak Modi. So, does vaccination prior to onset of pregnancy will reduce the placental infection, or there are any study yet to come?
It's just the hope that vaccination would reduce numbers of infections and hence vertical transmission. I don't think there are any studies. The problem being that pregnant women currently in India are not being vaccinated. There's just one paper which has come from the Pfizer vaccine, which has come, I think, only last week, which did show that the Pfizer vaccine is relatively safe. So we believe that even the Covaxin and the Covishield should be safe. So now the government is now thinking of doing the clinical trials in this direction. But since there is, I'm not aware of any single pregnant woman being vaccinated in this country, and we cannot have this data. I mean, reduction in vertical transmission is probably too far-fetched right now in the current season.
Okay. There's one question, last question I would like to take for Deepak Modi. It's, do you think that the SARS-CoV-2 could adopt a Trojan horse route for fetal-maternal transmission?
Quite likely. It looks like that it is doing that. There is now evidence for, there is some evidence happening, right? But difficult.
Okay. Thank you all. I hope there's no other new questions. So I will end the discussion session here, and I would request Dr. Vikrant Bo to take over the session.
Thank you, Nancy. And I think with this, we come to an end to a very interesting day. I think it was a scientific feast. And I think the amount of interest that it has generated speaks for itself. The number of participants and the questions. Actually, I mean, I was just following the variety of questions and the number of questions. That itself is proof that it was really very well attended and kind of successful event. And I will just now I have the very pleasant duty of thanking everyone, proposing the vote of thanks. So just to remind everyone that this mini symposium is being organized, was organized as part of our ongoing Department of Health Research funded training program for this year. It is titled Applications of Technologies in Genomics for Clinical Research, ATGC Research. So we were very fortunate to have this mini symposium as part of this program. So I would like to thank Secretary DHR and DG ICMR for the funding and for this opportunity to conduct this training program. I would like to thank our director, Dr. Gitanjali Sachdeva, for being accessible, giving all the approvals, even at very short notice. She was also very enthusiastic about it and all her suggestions. Thank you, ma'am. And it wouldn't have been possible without your support. I would then like to thank Dr. Deepak Modi. He's the actual force behind this program. And in fact, I had just asked, we had requested Dr. Modi to be in charge for this week of the course. And while contacting all the speakers, he came up with this idea of having this mini symposium. And within a very short span of time, we really got things very organized and contacted everyone and put that kind of infectious enthusiasm which only he can put. It's really nice to have. And thanks, Deepak. It was really a wonderful event, and I'm sure all of you will agree. So I think we should do more of it. And I would just like to add, we were talking about immunology and immunopathogenesis for COVID, and it was really good to see Dr. Sunil's work. And I would just like to add that we at NIRH also have been doing work on immune responses and immunopathogenesis right from April onwards. So we could bring out our reprint by the month of August, but we were stuck with our review of the manuscript, which got just accepted after a tough review of about 7 months recently. So I hope everyone will get to read about it. And it was
coming back. So it's fine.
Sorry. so you'll see more of it.
That's how it's okay. It's more of our satisfaction as well.
Correct.
Because
no, and I think we should be connecting. Yeah. It's good to know about your work, Dr. Sunil. So I would also like to thank the Prema's team from Prema's Life Sciences, Dr. Devjani Saha, Dr. Nityanand Sharma, and Divya Jyoti Singh for connecting us. And I think it was really, without your participation, and it wouldn't have been possible. You really ensured that we get the right speakers, all the right speakers at a very short notice. So a big thank you to all of you.
Thanks so much.
I would like to thank Dr. Ankur Sharma also for the wonderful talk. It was really nice to have him. Very rarely we get to hear about people from abroad at a very short notice, and it was very nice of him to accept our invitation. And finally, of course, Dr. Sunil Raghav, wonderful. Sunil, I have known you for some time now, if you may recall, and good to see you, and hope we can in future also.
So, sure, we should.
Yes. And of course, last but not the least, I would like to thank our IT team who made all this possible. They have been working full time, overtime. In fact, I would like to thank Mr. Ram, Mr. Ajay, Sushant, and Prathamesh for all the IT support that they provided. And also, I would like to thank our library and information officer, Dr. Prabhajit Kaur, for the publicity on the social media handles. And a big thank you to all the participants and all those who attended this event. It really made us very happy that it is well received. And we look forward to your participation in such future events. Thank you all.
And if everybody is okay, we can have the videos on and take a picture as a memory of this event. The participants, please, if you can switch on your videos.
Finally, Debjani is there.
Yes, you could see her. Tomrat, you think you can manage from there?
Yeah.
Tell us when we're done.
Thanks, Deepak. It was really nice.
Thanks, Sunil. Thanks for introducing.
Thank you so much, everyone. It was a pleasure listening to everyone.
Thank you, everyone. And
Today there is only one big sad news. Maybe everybody of you know now. From JNU. That was really frustrating and sad news.
Yeah, absolutely. I know. It has to be an end to this.
Yes.
Yeah, this is really getting too much.
All right.
Anyway,
thank you, everyone, and see you all tomorrow.
Bye. Yeah. Bye.
Thank you. Bye.
Thank you.